Good morning, good afternoon, and good evening. We have a worldwide group with us today, and thank you for joining us for the ASU Biomedical Diagnostics Educational Series. This is the second webinar of a new virtual series that the Biomedical Diagnostics program at Arizona State University is hosting in collaboration with Thermo Fisher Scientific. The theme of our session, which I hope you all know, is Diagnostics Technologies, the Lab of the Future. As everyone should be aware, this event may be live stream, photographed, or recorded. There is also a closed caption for those who need it on the screen. So when we talk about this session, as I mentioned, it's part of a larger series with Arizona State University and Thermo Fisher Scientific. And the core of it is all about diagnostics, the importance of diagnostics reaching their full potential for human health, clinical medicine, and research for the future. That is what we're all about. And from ASU's perspective, the College of Health Solutions is focused on better health outcomes require better solutions. And diagnostics, no question, is one of those important solutions. So with that brief overview, I want to, first let me introduce myself. My name is Mara Aspinall. I'm the co-founder of the Biomedical Diagnostics program, as well as a professor of practice in the College of Health Solutions. I'll be moderating today's webinar. And when we talk about today's webinar, we are going to talk about the future. But I want to be clear that we're talking about far more than just what to do with post-COVID labs and what to do with post-COVID capacity. We want to look out and make bold predictions about how labs will change, how labs should change. We'll talk about some trends that are optional changes, at least in the short term, but some that I think we're going to talk about the fact that they're going to be required in order for a lab to compete effectively and to be most efficient. So with that, let me introduce our three great panelists. I'm thrilled to have them all with us, and I want to emphasize that we have voices from a cutting-edge research lab, one of the largest commercial labs in the world, and the leading lab standards organization. However, they are going to speak from their own experience and their own background, not necessarily talk about, they're not going to be representing their employers. With one exception, Barb Jones. Barb is the CEO of the Clinical and Lab Standards Institute. Barb does speak for a large number of labs as the leading organization on the development and promotion of standards, education, and training to ensure that diagnostic testing has the integrity and confidence that is necessary around the globe. Barb has a lot of experience in the industry. Prior to her role at CLSI, she was Senior Vice President of Scientific Operations and Regulatory Affairs at Viant Bio, which is a drug discovery biotech. And before that, she has 25 years in operations, lab management, pharma standards, regulatory, business development at the national and international level. So we're thrilled to have Barb with us. Our next speaker is Stephanie McCalla, and Stephanie represents the area of research labs. She has a strong history, University of California, San Diego, PhD in biomedical engineering from Brown. She then moved to Caltech with a postdoc in chemistry and chemical engineering before starting her faculty position at Montana State University in Bozeman. She's got a broad range of research interests, most specifically for us today, focusing on microfluidics and molecular diagnostics. And last, but our third speaker is Susan Realegeno. And Susan is the scientific director of Quest Diagnostics. She is based in San Juan's Capistrano in the infectious disease department. Susan also has a strong background and scientific background with a PhD from UCLA in microbiology, immunology, and molecular genetics. She studied host immune responses. After her PhD, she was a postdoctoral fellow at the CDC, focusing on the pox virus and rabies branch. She has a lot of other experiences, has had the chance to be technical director for COVID-19 and molecular microbiology at UC San Diego before she joined Quest just a few years ago during the pandemic. And again, for our speakers, particularly Susan and Stephanie, they're speaking from their own experience, not necessarily representing the feelings of their employers. What I will say to all of you is that we are, as I stop sharing, as you could probably notice, we're a very large group, but we don't like to be in webinar mode. We are in meeting mode. So we can see, I hope, your faces. We want to interact. If you can be on camera, that would be ideal. But we want to have this as an interactive conversation. However, please, if you're staying on camera or not, please make sure your microphone is muted. What we're going to do today is the panelists. I will start with some questions for the panelists. We'll have some give and take. We'll get their approach to things. And then we will interchange that with questions from the audience. So please don't hesitate. I'm sure I don't need to describe this to you anymore, but put your questions in the Q&A. We will get to as many as possible. And at the end, I'll give you everybody's addresses if you want to follow up with specific issues. So with that, let me kick it off and start with a very broad question to each of our panelists. And the question is, can you identify one technology or scientific breakthrough that will become the prominent in clinical or research labs in the next 5 years? So what is that one piece that may be the tipping point for new technologies? So with that, I'm going to send it over to you, Barb, as our first speaker.
So thanks, Mara, and hello to everybody. If you had asked me that 6 months ago, I would have been all in on liquid biopsy. But I think that what the last 6 months have shown us is that AI is just taking the world by storm. And so I think that what's going to happen is that AI is going to be able to make sense out of this enormous assemblage of information around biomarkers. And what we'll start to see for clinical laboratories, that's what I generally speak for, is a shift, a real shift towards personalized medicine, where chem panels are, why are you looking at albumin for someone like me, a blue-eyed 55-year-old woman? Instead, maybe you should be looking at, osteocalcin or something like that. And I think that what we'll be able to do is not only that, but move laboratories into doing maybe population surveillance along with that, which will be very exciting.
So if we take that follow-up question, Barb, if I hear you right, AI is important, but it's a means to an end, not an end in of itself. It's going to, in your view, AI will be used in all the different parts of the laboratory. Is that fair?
Well, what I'm suggesting is that we have a body of knowledge around biomarkers that we simply can't make sense of. It's just the body of knowledge is just too big. And I think as we start to look at that body of knowledge on biomarkers or combinations of biomarkers, we'll be able to understand what we really should be looking at for specific people, because it comes down to payers. I won't go on, but I'll just say it comes down to payers. Everybody in diagnostics knows that it comes down to payers. And so if you can replace something in a chem panel with something that makes more sense and is specific to that person, I think that's where it's going to go first.
Okay, so we're going to remember follow the money, but we're going to come back to you and everyone and ask about the unintended consequences. So Susan, I see you smiling. What is your bid for the one technology that's going to make the biggest difference in 5 years?
Well, I definitely agree with Barb. AI is going to be huge, and it's going to, basically be applied to across all disciplines in diagnostic technology, and one of them is going to be metagenomic sequencing. Metagenomic sequencing, next generation sequencing has been around for many years, but it's been a little bit slow to adopt to the clinical laboratory for many reasons and hurdles that people need to overcome, and also more research in terms of clinical utility that needs to get performed. But I think, going back to what Barb said, liquid biopsy and metagenomic sequencing is becoming more and more prominent. More labs are interested in it, and they're trying to figure out ways about how to get more access to people so that they can use it, because the data has been shown that there is some clinical utility to it, and we're still exploring it. But I think metagenomic sequencing is going to be applied not only for diagnostic testing, but also for surveillance. We didn't really have we had strong surveillance programs for specific organisms in our country. For example, influenza surveillance has been pretty strong for the last few decades. But now that we see novel emerging pathogens, de novo discovery of pathogens and surveillance is going to be a big deal. And so metagenomic sequencing is going to be a way that we can do that.
So Susan, a follow-up question for you. If we stick with the follow the money, is that a big deal now because the technology has changed, or because the cost has gone down, or both?
I think both. So initially, yeah, there is a high cost, and now there's been more technology that's come out that's more affordable in a smaller scale, or for labs who are not doing high throughput. But at the same time, it's not only the cost of the technology and the bioinformatics that goes along with it, it's also the cost of reimbursement. Are you going to get money back for that, or are you putting in costs but not getting nothing back? So I think once more clinical utility studies come out, and there perhaps maybe CPT codes for it in the future, then you're going to see that pick up a lot more.
Okay, interesting. So it's both the cost the lab has and how much they're going to get reimbursed for it, obviously.
Exactly, yeah.
So we'll come back to, again, unintended consequences. But Stephanie, over to you. What's your number one?
So I actually was going to talk about sequencing as well, which I think dovetails really nicely into AI, as Barb was saying. So I'm going to, in the theme of, along the same theme, I would talk about nanopore sequencing. So that's one of the breakthroughs that has enabled this so much data, so much metagenomic sequencing. And it's because it used to be, obviously, sequencing was very expensive, and now we have these little nanopore sequencers that, around my PhD, there were electrical engineers saying, well, it's never going to work. And then maybe more during postdoc early in my career, people were saying, it's so dirty and it's so inaccurate, we don't want to use it. And now every year there's a paper coming out, it's better, faster, cheaper. And I think it's really going to start making a move into more diagnostics, not just the, transcriptome, but also, or not just coding RNA, but also small RNA. I think these same technologies, nanopore technologies, people are starting to use them in the literature to detect other molecules, small molecules. And so there's just so much information there. And I think along with that goes back to, so from what Susan was saying to Barb was saying, on you have all this data, what are you going to do with it? You can't just look at it and make a conclusion. You're going to have AI and machine learning. So you're going to need the mathematicians and the computer scientists, along with the chemists and the molecular biologists and the engineers. So I think it's this really exciting interdisciplinary technology that's all coming together that I am really excited to see.
That is great. So let me follow up with a comment and a question there, and then I'll start with you, Stephanie, and go to the others. 10 years ago, I made a prediction that labs would be hiring more bioinformaticists than lab techs. A, is that true today? B, will it be true? And how will labs, so it's a three-part question, how will labs deal with all this data that is being created, whether it's nanopore sequencing, AI models, or metagenomic sequencing? There's a tremendous amount of data coming out. How do they deal with it? Well, how do they analyze it with the current staff, and how do they deal with it once it's generated?
Yeah, and I think that that was a really good prediction that, you know, we're going to start to need more people in bioinformatics, more statisticians. I don't know if it's going to be more than, I think that Susan and Barb would probably be better qualified to sort of answer on that side. But I can say on sort of the engineering side and training our students, we do direct them towards fields like machine learning and computer science and AI when they have interests in the medical field, because it's just critically important to be able to understand the models and pull out sort of valuable information from an absolutely massive amount of data. So I do think there's going to be a lot of jobs in the medical field for people who started out in mathematics, computer science, electrical engineering, and that sort of thing.
So Susan, I see you nodding, and then over to you, Barb. Susan, what do you think, and how do labs, small and large, deal with this amount of data?
Yeah, I mean, I definitely agree with Stephanie. They're going to be way more important as we go on and start using these technologies and more. I think our field is a little bit slow to pick up on trying to get more bioinformaticists, but there's also a lot of companies that are providing solutions to people who are not bioinformaticists and also laboratorians. So it's more of a user-friendly interface that they can input the data, and then it could do behind the scenes analysis that has been validated and tried, and then give an output that can be easily reportable by like a clinical lab scientist in terms of the amount of data and how to handle it. So that's actually a big challenge. So one, there's a lot of regulatory and compliance around it where you have to secure the data. And so you have to find either a cloud-based or a local solution where you can store that large amount of data for several years, because you have to keep the data for at least two years, I believe. And so that needs to be in a place where it's accessible and it's not, not widely available to people outside the laboratory or the institution, and also that can hold a lot of it. And so I think that's probably one of the challenges. So the more cloud-based solutions that are secure, that are going to be out there, I think that's going to be a way that people are going to go for storing their data. And then also an easy way to pipeline the data storage with the analysis and with the reporting. And so having that infrastructure from start to finish for the post-analytical phase is going to be really important. And maybe you're going to have to piecemeal it for now, or but once somebody comes out with a full start to finish, that's going to be very popular.
So that's a perfect transition to Barb, because Barb, my understanding is you can't just use your sister-in-law's web finder or cloud storage. It's got to be HIPAA compliant. It's got to meet some standards. So broadly, how is the field talking about it today? What is CLSI doing about setting the standards for this type of data in the future? So I'm going to add a commentary here. We continue to have trust in labs to hold and store this data.
Right. Well, before I get to that, I just want to say that I see this as a huge opportunity for labs. I don't want this to get missed, because up until this point, labs have been the information hub for healthcare, but they don't act like it. They don't step into those shoes. So as I think that there's a statistic that people throw around that 70% of medical decisions are made using laboratory data. I believe I looked for the study. It's a small study that's being referenced. Nevertheless,
and like 15 years old, but it's still true.
I believe that it's it's increasing, but the laboratories haven't stepped up. And as we start to add in real world evidence, which gets to what kind of data do you incorporate and where and how, then the laboratory can become this informatics hub. And so just to everybody out there, if you are running a laboratory, get this capacity and step into those shoes, because you rightfully belong there. In terms of standards for this, CLSI isn't the information standards organization. There's an organization called CDISC that's out there helping everybody to understand interoperability and how you work with those standards, but what we do, or what we will be looking at, is the use of AI across the board in determining and making decisions that lead to clinical outcomes and clinical decisions. So we'll be looking at that, but we need a lot of help in that area. It just came on us like a hammer, all of us.
So a question from me and a question from the audience. We don't have to wait till the end, so let's integrate those questions. Barb, is your sense that 5, 10, 25, 75% of labs are already starting to work with AI systems? Where are we on the 9-inning? I'm a football and baseball fan. Where are we in the 9 innings?
Yeah, so I would say the new generative AI, nobody's really incorporating to the extent that I think it should happen with bioinformatics and taking sort of data and figuring out, oh, we have a vitamin D deficiency in our area, we should start screening for that. None of that is happening, right? But there is some automation and smart laboratory information systems. It's already using machine learning to help guide labs in the right direction in terms of reporting and deciding what should and shouldn't be done. But I think there's a lot of work to be done, and I really hope people start to embrace that surveillance and generative type of exciting opportunity out there.
So we have a question from Professor Travis Schlappe. So I'm going to ask him to unmute and ask the question live. But then after that, to give you some sense, I want to talk about unintended consequences of this AI and what dangers there are out there. So Travis, over to you.
Hi. Yeah, this one's for Barb. You sparked some curiosity with your statement that labs are not the information hub and they should step into that. So who is the information hub right now? Or is it just dispersed everywhere and there isn't really one?
I shouldn't say that all labs aren't information hubs. There are labs across the world that have really taken that mantle and run with it. It's a respect thing. I'm sure that those who are in the audience who run laboratories understand what I'm about to say, and that's that it's seen as sort of information gathering rather than information processing and delivery. And so it's being left to different clinicians to be able to take all that information and make decisions. That's not to say that laboratories aren't hinting at things for people, particularly in the microbiology area.
Super. So thank you. Another question for everybody from Wendy Diller. I'm trying to find Wendy. Wendy, can you unmute, or would you like me to ask the question?
I'm here.
Great.
Thanks.
So why don't you ask it?
Yeah. Okay. Thanks, Mara. What kinds of technologies and vendors will labs need to work with to incorporate to become stewards of AI-generated data? And also, labs generate a lot of different labs in other parts of healthcare generate a lot of different kinds of data. How would labs be the stewards integrate that data coming from different areas? Or are you referring, those two different questions, or are you referring specifically to lab generated data when you talk about labs being stewards?
Barb, you want to start, then we'll go to Susan.
I do, because this is really important, because the 21st Century Cures Act has broken down, or should have started to break down, barriers in terms of interoperability and data sharing and patients having access to their own data. And I think that what we'll see is also an insistence by the healthcare consumer that their real world evidence start being incorporated into their clinical plans. And I hope that happens. So I believe that it's going to be a both way thing. It can be laboratory generated data. Of course, you have point of care testing data that comes to you from sort of satellite to the lab. And then you should have also real world evidence. Right now, it's very disparate in many laboratories, in many hospital systems.
So Susan, let me send it over to you and take it from there. Although Barb, if you have a moment, you mentioned an organization that's setting the standards for this, and if you can put it in the chat so everybody knows what the organization is, that would be helpful. But Susan, over to you. What kind of technologies and vendors, large labs, small labs, are labs going to create this themselves, or they're going to use a third party to do this sort of AI analysis?
Oh, I think they're definitely going to use a third party, and there's already a few companies out there that are applying their AI technology, and it's being integrated in, for example, total lab automation, where, you know, pictures are being taken of plates, and then they're sorting out negatives or positives and saying you should look at this, don't look at this. So I think they already exist, and they're being put into different technologies that the laboratory is already using. And so it's going to be a matter of the voice of the customer, you know, telling what the vendors and people who are developing these technologies, what the needs of the lab are. So if you look at your specific lab, whether it's small, large, and you say, me as a small lab or me as a large lab, we need these things. And so, you know, speaking to vendors, particularly at conferences, they're very open to, you know, input from their customers. And then they take that back and either prioritize it or figure out how they can, they could be, you know, they could incorporate those technologies into what they're already working on so that they can, you know, cater to all their customers. So there's going to be some vendors that want to focus on small labs and maybe point of care or maybe low throughput, and that's going to be their market. And there's going to be larger vendors that work with larger like laboratories, like reference laboratories, and find ways and solutions that could be implemented across several laboratories and also who have high throughput volume and able to take that data. So I think it's definitely going to be a collaboration between laboratories, their needs, and voicing that over to people who are already working on it, and commercial vendors and manufacturers and also researchers. So, you know, knowing the researchers already working on that and getting it to a point where a manufacturer can take that research and implement it into a commercial product.
So that is really helpful. I assume it's a lot of let the buyer beware to make sure that, you know, the hundreds of companies saying we can help you with your AI, to make sure that they are qualified to do so. But Susan, you mentioned the research labs. Stephanie, let me go over to you. Are you seeing the exact same phenomenon? How is it different from a standard clinical lab to a research lab?
Yeah, I mean, I would say it's very different. And this is something I think that is going to come up later. But for us, by the time we get data, it's often de-identified. And with the labs like Quest, you have a bunch of data that falls under HIPAA. And so I think that we can, you know, have a lot more people working with software they maybe created themselves, or their collaborators created themselves. I know that there are a lot of small companies popping up to do this type of work because, you know, we send our students out into the world, into those types of places. But I feel like for us, there's a lot less regulation and pressure on how we handle data, as long as it's not, you know, identified patient data. So different
you said
world.
The magic word there, de-identified, and probably regulatory, because you've got far fewer regulatory issues. So
We do.
Let me take that on the unintended consequences. Samantha Berg, I'm trying to find you, but if you're there, can you unmute and ask your question? Or if I don't find you quickly, I'll ask it.
Hi.
Wonderful. Thanks, Samantha. Go for it.
What are the regulatory concerns with like unsupervised machine learning? Because we don't know what it's training itself on, what biases it might be introducing. And then also because a lot of AI is being used to do functions that humans straight up can't, there's no like ground truth to compare it to and to use to validate for clinical use. And also currently the software validation recommendations for a lot of regulatory bodies are really vague. So are there things, do you anticipate things coming down the pipeline that'll have clear guidance on validating AI for clinical use? Or do you think this is going to be a very difficult sphere?
Barb, you want to start with that one?
Yeah, I think it's all about the standards, right? NIST, the National Institute of Standards and Technology, is really spending some real effort on validation and standards around the use of AI. So I think that will, help set a foundation and a baseline for everybody. My daughter, who's brilliant, explained it to me this way for generative AI. She said, you're not necessarily getting the true answer, you're getting the answer that another computer model can't identify as false. Wow, big difference.
Yeah.
Big difference. And that blew my mind. So you're exactly right that we need standards around it. And as I say, Nara, I'll just end here saying we're not doing it at CLSI, but certainly NIST has taken on this mantle for all AI across all areas, which use the same models.
Wonderful. I appreciate that. And that sort of answers Martin Ramirez's question about which of the government organizations. I'm going to take one more question. Jose Hackett-Murgas, if you're there and want to unmute with your question on AI, and then I'm going to move on a little bit because we've got a few other topics. But this is great.
Yes.
Let's keep sending questions so we can have it an interactive. Jose, over to you.
Yes, hello. So I have, it's a little bit of a two-part question. The first part was about like how far the reach, we're all talking about here, like incorporating AI into diagnostics, into like however that integration would be. I was wondering a little bit more on how far that reach should be into that, like how far the reach of AI should be into like creating those like prediction models or creating more of a model regarding like how much of that AI readable information should be taken into consideration for a model. Yeah. I mean, I work in a clinical, like commercial laboratory. There's a lot of data that can just be taken at face value. There's more or other information that a lot of times is not, as clear that we can take. And the other part, it was, we're talking here about both commercial laboratories and research laboratories, how far that collaboration should go.
Great questions, Jose. Complicated ones, obviously can't be answered very quickly, although I'm going to ask our panelists to answer them very quickly. And if we can, we'll get back to some other questions on AI, but after this, we'll move on a bit. Susan, Stephanie, do either of you want to start on the collaborations between research and clinical labs, and how far can AI be, or at least what do we need to look for to set some parameters? Stephanie?
Yeah, I can go ahead and try to tackle that one. You're right, those are wonderful and obviously very complex questions. As far as how far AI should go, I think I agree that there is some concern there, but we're talking about data sets, at least anticipating data sets that are so big that there's kind of no other way to deal with them. So I think having maybe not AI the way people think of it, like ChatGPT, like generative AI, but having things like a machine learning to pull out the most important parameters in models is going to be pretty important. But obviously there is going to need to be regulation on.
Yeah, and Stephanie, I use that as where we started, a means to an end, using AI to help inform the research, not do the research.
Right.
To me is what I hope is the short-term focus. But Susan, where are you on this?
Yeah, I think one thing that's really important to remember is that we have to identify what the need is and how AI or predictions can help fill that need or what's missing. and then from there go on to validate it. Is it, you know, as good or better as our gold standard, what we're currently doing? And so even though we have so much data, you know, I think, you know, part of it is also collaborating with researchers and kind of looking at how we can better use that data and what it actually means. But once it starts getting implemented in the laboratory, it's really important to already have it validated in your process and know that what you're using is for a certain purpose, and you're not going to misuse that information, or there's not going to be a risk of that information being widely misused, or people are not going to know how to handle it. So I think it needs to be really well thought out before it goes into the clinical laboratory. And in terms of like collaborating with researchers, I think diagnostic labs actually need to do that a lot more. I think that, you know, there's a really good partnership that could be started there where, you know, the diagnostic space does have a lot of needs, and working with researchers to co-develop some of those needs and actually, you know, either providing de-identified samples or data or information to them that they could use to improve either on whatever methods we're currently using, or develop, you know, AI solutions for some of that data that does exist, and like I said, have already a purpose in mind about how it's going to be used or how it can be used. So we're not kind of blindly using it. And that's very different from what like Barb is saying, where we have a lot of data out there. How can we bring it together to kind of look at population health and better guide like where the needs of healthcare can go? And so I've already seen people starting to do that at small institutions where, you know, they look at, you know, it looks like there's a high rate of hepatitis C in this population. Let's do more testing in these areas, or more education. And so I think that's where a lot of the data and how it's used might go in terms of population health, and looking at all these different sources to kind of paint a bigger picture and kind of guide resources to where they're needed.
Super. Well, Jose, thank you for the questions. Great answers. Let's move on, because we won't be just about AI today, into, if I had to do a transition, I'll use the word trust. And that, Susan, you implied, one of the biggest shifts that we've seen during the pandemic is the use of home testing. Many of us for years and years said it's going to shift to the home, it's going to decentralize, we're going to have lots of testing at point of care and home. And quite frankly, it didn't happen until COVID-19. And even in the first year of the pandemic with home tests, it didn't happen until it became a crisis. So then home testing was established, at least for COVID. Mixed blessing for labs, I would think. One is it's taking it out of the lab, which is both good and bad news. So I'd love your reactions to what, you know, is home testing here to stay? Or was this a blip because people didn't want to wait online for a lab test? Or is this something that fundamentally changes the way we get the information? And either way, how do clinical but also research labs react? Barb, I'm going to start with you on this one.
Oh, this is my favorite subject. I could talk about this for 400 years. But
don't worry, we won't be here for 400 years.
No. I think healthcare consumerism is on the rise. I think we're seeing it everywhere. And you're seeing what I call, not in a derogatory way, but absolutely, I'm excited about strip mall lab testing. So consumers that go in and they want their own panel for health optimization. And I am so excited about this. I started my career as a dietitian, trying to help everybody take control of their own healthcare. And so this is very exciting for me.
But what about the risks?
I'm sorry?
What about the risks? What, you know, giving 1 million people the test to do it at home, what could go wrong? What could go wrong?
What could go wrong? Well, you know, I think that it is about trust. And I personally think that we grow as healthcare consumers over time. And what you'll see is a proportional growth of information and the ability to deal with that information with the growth in healthcare consumerism and at-home testing. So for right now, you have yes and no's. That's hard to screw up. Yes, I have this. No, I don't. Or yes, I think I might have this, or maybe I'm still worried about it. It doesn't matter. It's a yes and no. But when you start to get things that are like continuous glucose monitoring, where you get values throughout, and you have people making decisions, you're going to have to see an increase in education as well. But I think it's happening. I'll stop there and let others, I'd love to hear what others have to say about it.
Yeah, Susan, I see a lot of nods, and there's a question in the chat. How much are we willing to trust home testing with other diseases? What about false positives? What about, today insurance companies are saying you got to go do the test again. Susan, where are you on this home testing, and what should labs do about it?
So I think it's inevitable. I think at home or outside the clinical, like the clinical hospital space, this is going to happen. It's going to be a matter of how do we reduce the risk to the consumer and link them to care. So a lot of the risk is in in specimen collection. Are they doing it properly? Are you increasing your risk of a false positive or a false negative? And then the second component is if they're performing it at home, same thing. Are they doing it correctly? And so reducing the risk there is all going to be about education, making sure that they understand how the testing is supposed to be used, or what to do with a certain result, what actions to take. And then also if it's being sent to a laboratory, knowing that, that there's linked to care when they get a result back. And so I think a lot of it's going to be education, reducing the risk, and then also false positives and false negatives. It's all going to be linked together. Telling the patient this is how you properly do this. If you think that you're not doing it properly or you need a confirmation, go see your provider. At the end of the day, I think it needs to be a provider-consumer relationship, and I agree with Barb. People are being more aware and taking their own health under control and wanting to know more. I mean, before COVID-19, people really don't know the name of viruses, what an antigen or a PCR test are. And now they're asking for CT values, which is really bizarre. And so, what that means and how they take that information, we need to make sure that we educate and mitigate the risks to the patient. So make sure that the risks are as low as we can make them, so that there's no negative effects on how they use that information. And so linking them to care is going to be really important.
I love that. I do have to comment. I heard somebody say that before the pandemic, people didn't know the difference between PCR and CPR. But the fact that they're asking for CT values and we're debating antibody levels, is pretty extraordinary. So we're going to enter into our last 10 minutes here, and then I'll do a wrap up, which I'd like to call the lightning round. And I'm going to start with a broad question that has come up a few times. And Michael Donovan, you mentioned it in the chat. Can you ask your question about laboratorian shortage and staffing labs?
Sure. Thank you, Mara. How are clinical laboratories and even research laboratories, from Stephanie's perspective, handling laboratorian and research laboratory worker shortages? I know we talked about AI quite a bit in the first part of this discussion, but that seems more like a long-term solution. How is it being solved in this week, basically, from a urgency standpoint?
And let me add, I believe that the average age of a lab tech is in mid-40s, and the average age of a histotech is in the mid-50s, which is not typical of the rest of the healthcare industry. Who wants to start there?
I can do that
Go
one.
for it.
Yeah, because I talk about this all the time. That is a huge fear of what's happening for at least one of our constituencies, medical laboratories. And the way they're handling it is automation, definitely automation, and making the human being be the quality check. So reducing the number of people on a line, but having those people more responsible for quality, and pushing a lot of the point of care testing out. So the nurses are performing a lot of testing at bedside if they can, and training others in the hospital setting. But society is handling it differently, in that we're doing a lot of testing in different places we never thought we would. For example, a pharmacy
Yeah, or a school.
or a
I mean,
school.
in
Yeah.
a big way. So Susan, how do you think about this? Give us your perspective.
Well, I mean, going back to what Michael was saying is like, how are we going to deal with it today and not like 5 years from now? I think one of the things that we do is we look at small little steps of it. You look at, your post-analytical and your pre-analytical and say, what are small things we can do here that we can automate? Is it, taking this one step and finding a solution for it today so that we can reduce in that the hands-on time and that person can go and do something else. So I think those are the immediate solutions that a lot of laboratories are looking at, how to use tools that we already have and just increase those tools so that they can automate steps that are repetitive or do not necessarily need any calculation or like thinking or just things like automation, like aliquoting or pipetting. And that capital is a lot smaller than AI. And so investing in those, small little things kind of add up. And so you do see workflow efficiencies, just, putting automation in just one component of your workflow. And so that's one thing. But another thing that we really need to focus on is training now and encouraging people to get into the field because we're going to really need them. And so, encouraging people to go into these training programs and, training them for the needs of the laboratory, I think it's going to be really important.
Super. Thank you. Thank you for that. Let's move on to, hopefully we can get in one or two more questions. What about, I said it's not just about COVID and excess capacity, but what growth opportunities are there in a lab today? What are the areas that you think are going to grow in terms of lab testing in the near-term future and how best to do that? Stephanie, let me start with you in terms of research labs. Are there new areas of research that can't be done at home and that really need to be done and labs should be thinking about adding them?
Right, yes. So I do think, I guess I'm a little biased. My research interests are in point of care and sort of simple diagnostics. So I do feel like that is sort of technologies that are up and coming and good to it's not just things that can be, a lot of the things that can be used for clinical labs can actually also be used for research labs. So especially at smaller universities, there might not be a lot of startup. I have a lot of colleagues who are in the Montana University system who are on small campuses. There's not a lot of infrastructure. So having a very cheap way to do PCR that they could give to all their students, and then their students can, rural healthcare is also a big issue here. So can they go out and set up some small labs in more rural areas? So I think those technologies have a lot of overlap between what you would need in research and education, as well as in the clinic. So any technologies to simplify things like detecting DNA or RNA that you can move into both small, clinical labs in the middle of rural Montana, but also in, small teaching universities, I think could benefit both.
And Stephanie, that's helpful because I was actually going to ask about whether the footprint of a lab changes. And what I heard from you to say is we may, particularly in rural areas, have more smaller labs rather than one mega lab because research or clinical. Susan and then Barb on this question.
Yeah, I think with, the excess capacity and all these instruments that are around, there's a few things that laboratories can do. One of them is create partnerships similar to what Stephanie was doing is basically finding where there's people who need the need for those technologies and creating partnerships so that you can share that technology with other institutions or with other partners. The other thing that laboratories can do is develop testing that they don't normally have before. So now you have all this excess capacity that they use for COVID. What else were you not doing before that you were sending out or that you would like that you can develop either more laboratory developed tests or bring on other testing that could improve patient care by increasing your turnaround time or doing it in-house. And, one of the challenges there is that you need expertise to do that and people. So I think it's kind of a give and take here. So, but
you may have to prioritize depending on the people you have.
Exactly.
It all goes back to the people. Barb, your comment here?
Well, I think probably sequencing, everybody should be doing it. Drug companies are increasingly tying drug approvals to a genetic marker. And so the sequencing is just a requirement, more and more and more. And so I think genetic sequencing is a big deal. And then I would add to what Susan said, which is that doctors' offices and doctors' office networks are creating laboratories around their work so that they can decouple the laboratory testing with the physician visit, so they can have people go in and get testing before they come to visit and talk to the clinician with laboratory tests in hand.
Yeah, I appreciate that. And sequencing, as we've heard, the broadest definition from including PCR, nanopore sequencing, and other types of sequencing. So for our very last comment, then I'll do a wrap up. I see Ted Snellgrove had an interesting comment and Tradeoff. Ted, can you go off mute and just ask your question and add a comment?
Sure. Good morning, Mara. As the metagenomics field comes together enabled by AI, it will allow much more precision use of the most expensive therapeutics, which will in some ways significantly limit days on therapy compared to current practice patterns where they treat till radiation or radiology declares some kind of victory. In that world, it's going to change the model for how pharma doses some of their expensive drugs. It may change how FDA adapts as well to this kind of opportunity with precision data on each case. I'm curious if there's any thoughts about how pharma and FDA will adapt to this changing information space.
So I'm going to have to ask very quick answers here so we end on time, but I think it's an important point that we need to think about for the future. Susan, Barb, Stephanie, any quick comments on that? Do you generally agree we need to be looking at this issue in terms of a trade-off of embracing this trend?
I think there's already discussions on it. I think the FDAs are really thinking about how we're going to validate this, how are we going to have oversight over this, and how do we decrease patient risk. And so the discussions are happening. I think it's just going to be a matter of, coming up with a set of standards that people can follow in order to make sure that what they validate and what they approve is high quality. And so yeah, that's going to be really important. And, thinking about it now before it's at the forefront. You don't want to think about it after the pandemic or during the pandemic or while it's happening. You want to think about it now and kind of predict what we need.
Get it in early. Stephanie, you agree? And then I'll give the last word to Barb with that word, standards. Stephanie, thumbs up to agree with what Susan went.
Yes, I completely agree. I think the conversations are already happening. It is something that I'm glad that a lot of people are chiming on because obviously when you collect that much data, like who knows what you can kind of back calculate about people, who knows what type of therapies that's going to open up. So I know the conversation, I'm grateful that they are.
Super. Barb, 1 minute or less.
Yeah, I'll just add that I hear the word standard and I get all excited. Yes, absolutely, standards.
Love it. Love it. And thank you for posting on the chat the other standards organization that you had mentioned. So with that, I am very pleased to share my screen and wrap up. Let me thank our great panelists. You did a great job, and you'll get their emails in a moment, but really by being both provocative and practical, which is exactly what we had hoped. Let me tell you about the next discussion in this ASU Thermo Fisher Scientific series. And our panelists today highlighted it, the need for education. And we talked about lab and patient education, but next time we're going to talk about physicians, healthcare providers education. And just like today, we have a great panel coming up, and you can see the names here. And I will not break any HIPAA laws, but I will just say one of these panelists is actually a patient and talks about how he had to deal with a very tricky situation as a patient in terms of educating his physicians on the challenge of diagnostics and why and how to use diagnostics. So please join us, same time, same bat channel, November 15th, 8 a.m. Pacific, 11 a.m. Eastern. I'm also pleased to say our annual ASU Diagnostics Summit is almost upon us, virtually 30 days from now, Thursday, October 19th. Please, please, please join us. 8 to noon, we'll have a great discussion. Tim Stenzel, as everybody on this call I'm sure knows, the FDA Diagnostics Chief, is going to be our keynote speaker. We will continue the discussion on AI. We'll continue discussion on home is where the test is, and with public health officials talking about that. And then the topic that we also talked about today, follow the money. Diagnostic reimbursement, the good, bad, and ugly. Let me know or follow up on this. We'll get you an invitation and put you on your mailing list from noon to 1 o'clock that day, in person and on Zoom. This is a hybrid event. We will be having a special session with a team from ASU Health. So we're thrilled to have a very exciting, again, interactive session. Join us on October 19th. So as we summarize, I do have to say at ASU, we have a master's degree in biomedical diagnostics. It's 1 year, it's online. And with Michael Donovan, Travis Schlappy, and so many others, it is a fantastic program. Go to our websites to get more information on diagnostics, particularly COVID. And when we talk about diagnostics at ASU, we're part of the bigger college, the College of Health Solutions, which has programs in diagnostics, biomedical information, healthcare systems, nutrition, population health. So CHS is the place to be. So with that, let me just say thank you again to our great panelists. If you want to follow up, they have given permission to give all of you their email address. We'll be following up. A recording of this will be available. Thank you all, and hope to see you at our next series. And we're happy to have comments, feedback, good, bad, ugly. Tell us what we can do better. Stay well out there. Thanks, everyone.